Excellent. Hi, everybody. So I sort of realized that about 10% of you are not awake yet. And if my voice can't make it, just go to sleep or be ready for the next speaker, who's probably better than I am. I will tell you not a little bit about basic biology to biotech, because over the past 6 years, Anders and I have built a company, Orphazyme, and we're now 25 employees in the company. But essentially, the first title of the talk was something with our Lego blocks, genes, or genes Lego blocks. And from that, I sort of tried to think a little bit about what that actually mean. I talked with Anders about what did he really mean about that. It set my mind into deconstruction, and that's a general theme that I'll be coming back to, and reconstruction of diseases as well. Basically because I realized that the topic of today is the future of life science, which of course involves a lot of you people eventually down the years, but also because there are some mega trends that are happening right now in science, and particularly in the pharmaceutical industry. And we are sitting, with Orphazyme, we're sitting sort of in the middle of that and getting to talk to a lot of companies about what their focus are and what they want to have from new and developing companies. So I'll try to relay that a little bit to you, but using Orphazyme as a case example of how you can deconstruct a common disease, in our case cancer, and then reconstruct from the rare disease it went into and back up into another common disease, which you probably know, it's called Parkinson's disease. So with that, we'll just start out. As I said, we'll be doing a little bit, it's rather sensitive. We'll be doing a little bit of cancer deconstruction, which was basically the foundation for Orphazyme. As Christopher pointed out, I was working at the Danish Cancer Society. I did my PhD and my postdoc there. So just very quickly, how many of you are PhDs? That's a vast number. Postdocs? And master students? Excellent. Right. Okay, I also did my master's at the Danish Cancer Society. And basically, it's a long and arduous process. And what we worked with at the lab of Mariette back in the Cancer Society was deconstructing cancer, basically. That's what everything is about in understanding diseases, at least from where I come from, which is with a biochemical and human biology background. We want to understand diseases at the molecular level. And so we tried to do at Maria's lab, and what we were really interested in terms of cancer progression, was how the hell do cancer cells avoid cell death? They should be dying. I mean, the body is sending tons of different signals and cellular signals to these cells, and still they keep on fighting, still keep on living. And we wanted to understand the processes involved in that. With that, we started to look into different things about cancer, and this is Maya, and she still has a group, and she's now the leader of a huge department down at the Danish Cancer Society. She's fantastic. If you need a place to go and talk with a really good professor, go to her. That was my advertising for Maya. Things started out a few years before I did my master's thesis. So we had observed back in the lab that cancer cells, if you deplete them of this strange protein called heat shock protein 70, which is a molecular chaperone, then essentially you can activate tumor-specific cell death programs. But what was rather curious, and what we spent a lot of time understanding, was it completely bypassed caspase activation and BCL2 antagonism. And for those of you who are not familiar with cell death, that was the hype in the late 90s and early 2000s. Caspases, apoptosis, and caspase-driven cell death was the way that cells were supposed to die. If that didn't work, you would have necrosis, and those are sort of the two outer points in understanding cell death. Well, whatever we did, if we deplete these cells of heat shock protein 70, they certainly didn't die by caspase activation. So we tried to understand what was going on in these cells. And we fought, in particular Maya fought a long and hard battle against the establishment, based on the data, basically, in trying to understand what was happening. And we figured out eventually, together, that was Jesper's paper back in DXMed in 2004, that what heat shock protein 70 actually does is that it promotes cancer cell survival by inhibiting what we call lysosomal membrane permeabilization. Within our cells, we have small vacuoles called lysosomes, which are organelles used in recycling of macromolecules. In order to do so, they have a number of proteases and hydrolases inside them, and if they start to leak, it's not very good to be a cell. They will die from it. So what heat shock protein 7 did was basically to help cancer cells survive the stress of transformation and keep the lysosomes functioning. eventually my project was, when I came into the lab at exactly this point in time, or a year before, Maya sat me down and told me that I could have a project which would probably be very difficult, but if it didn't succeed, she would help me anyways. And that's always very good to know, that your mentor is willing to take care of you if that strange project that you're pushed on doesn't really work. But 6 years later, and with a lot of frustrations, we ended up with a success, essentially. We understood at this point how heat shock protein at a molecular level stabilized these lysosomes. And that was basically all I worked on for 5 or 6 years. No publications until then, and of course it's very good then to have a good publication, and that also significantly helps you if you want to start a biotech company, I should say. Now that was one part of the story. The other part was, if I can get this to work, was another thing that came out of it, which we published a few years later. And that is, you could either use heat shock protein 70 and the mechanism we discovered for this very strange disease called Niemann-Pick disease, which nobody really has heard of. You could also use the same mechanism and turn it around and basically start to kill cancer cells by exploiting this mechanism. So the big question, the question I very often get is, we went off with Orphazyme, of course, and this is from Nature Biotech back in 2013. We started working with lysosomal storage diseases. That's the whole foundation of Orphazyme. And the question I very often get is, why the hell did we choose rare diseases over cancer? And there are a number of very good reasons for that. I'll try to go through them. There are business reasons, there are personal motivation reasons, and there's sort of just what was happening in the field at the time. But this is about 6 or 7 years ago now. And if we take it from the business side to begin with, a lot of people don't really appreciate what rare diseases mean and the incentives put in place to develop drugs for rare diseases. Now, this is the poster story of rare disease development. This is Ceridase, the first treatment for Gaucher's disease type 1. It's a recombinant enzyme. This is not a recombinant enzyme. This enzyme was purified from placentas. I think they used about 18,000 placentas per vial for injecting into patients. That was a huge, huge, huge undertaking, and nobody really understood why the hell this little corporation at the time, Genzyme Corporation, was going for this. Well, as it turned out, there we go. Genzyme has ended up selling Ceridase and later on Cerezyme for about $1 billion per year, fluctuating between $650 million, and I think they ended up with $530 million for the first half year last year. So of course, $1 billion is sort of makes people look. And this is a rare disease. We have about 8,000 patients in the world suffering from this disease. 4,500 in the US that are registered, whereof I think 3,000 and 3,500 are on this compound, 4,500 throughout the world. Another thing that was also happening and which sort of caught people's eyes was this product, Soliris, produced by Alexion for a disease called paroxysmal nocturnal hemoglobinuria. It was a few years ago the most expensive drug in the world. It has been surpassed since, but you can see what you can demand in terms of pricing for an orphan disease product. $400,000 per patient per year. That does add up, even though you only have 1,000 or 2,000 patients. That's the business side. That's why investors are keen to invest in companies like Orphazyme and other companies that are dealing with rare diseases. That, together with the incentives put in place from a regulatory perspective, the FDA came out with what is called the orphan drug legislation back in 1983, 17 years before Europe, where Europe introduced the same legislation, providing a number of incentives for companies to develop drugs for rare diseases. One paramount incentive is that you are essentially competition-free for the first 7 years in the US and 10 years in the European Union. So if you go first to market, you have market exclusivity, unless somebody comes up with a superior product, but that's very hard to produce within that time frame for a very rare disease. We're basically also controlling the market as being the first mover. So there's a huge financial incentive to develop drugs for rare diseases. The other one, of course, if you really want to do this, you need to have some sort of personal motivation. And I tend to stress this when I give other talks to students about why should you go into building a biotech or do something with your biology, with all the things that you've learned and the things that you're studying. That is to find some sort of motivation. These are the cells that we looked back in November 2007. And I still remember this experiment. Some of you might have heard this story before, but we are talking the mid of November 2007. I was rather fed up with everything. I particularly hated cancer because I was tired of working in culture systems that I thought had absolutely no resemblance to the human conditions. HeLa cells, U2S, DU155, MCF7, you name it, they were rather boring, to be honest. They're probably good for something, but at that point in my life, I thought they were not as interesting as they could have been. So I got hold of some patient cells. At this point, we had discovered that heat-shock protein 70 works by stimulating acid sphingomyelinase. So I took a look around and looked at genetic diseases where acid sphingomyelinase was deficient. And then these Niemann-Pick diseases popped up. Niemann-Pick disease type A, B, and C. And when I looked into those diseases, they didn't have any treatment. There were a few thousand patients scattered throughout the world, maybe 8 to 10, just like Gaucher's disease. But essentially, they don't have any treatment whatsoever. These kids are dying from the severest form before they're 2 years old. If they're lucky and they will have the less severe form, they will still be dying and suffering tremendously while they develop the diseases. So I was sitting with a dish full of patient fibroblasts, which were taken at passage 4, which means they've come from the patient being passage 4 times. And that's basically as close a system as I could get to the patient. And I felt rather invigorated by that. So what happened was that we put HB7, the recombinant form of heat-shock protein 70, on these cells. That's what you see in green. The red cells, I'm just going to give you a little bit of lecture in cell biology. These are cells, individual cells. Cells that have taken up heat-shock protein 70 are green because we labeled HB70 green. Now, as you can see, a cell that's not taking this up is completely filled with red, which are lysosomes. And that's why these diseases get the name lysosomal storage diseases. They're basically filled with non-functional lysosomes. If you put out HB70, lo and behold, you basically revert back to normal. I ran to get Maya when I saw this, had her blinded, sitting down at the microscope, and I was lucky because she was in, it was a late Wednesday night, but she was in working late and so was I. And she sat down at the microscope and looked up at me and said, "Really?" And all we could do is, yes, I mean, that was it. Those were the data. So we sat with something which we both found to be very profound. And that's essentially, this is the basis of the Nature paper. Of course, all the mechanistic world of work led up to this, but the major discovery was that we could do something that we thought was really transforming the molecular pathology of the disease. When you sit with that, there's 2 ways, or for me there were 2 ways to go. One was to go with the Nature paper, stay in academia, go abroad, go to US, go to Harvard, Stanford, MIT, wherever you wanted to go basically, do a postdoc, come back, and start a group. The other was that I always wanted to start a biotech. And this was a little bit premature to what I had planned for, because I had planned to go abroad, come back, start a group, get a good idea, and build a biotech. It sounds rather simple, right? I don't think it is, but anyways, we had those data, and I also realized that there's nobody to move this unless you move it yourself. And I think that's the most important take-home I can give you. It doesn't matter how big a discovery you make, you need to move it forward. And there's only one person who can do that, it's you. The personal motivation is absolutely crux to everything you do and all the interactions you will have if you want to start a company and move it forward. It is your energy and energy that you put into it together with your co-founders that are really starting to move things forward. So if you consider doing it, please do it, but just realize that it will take your energy. You can't sit on the sideline and expect things to happen for you. Anyways, here we go. Saturdays, and now I'm going to go from rare to common. So I hope you followed me on how we took cancer, understood the molecular mechanism of cancer, and could use that to treat a rare or a group of rare diseases, in fact, the lysosomal storage diseases. What happened in the meantime was that everybody was starting to look a little bit more interested at lysosomes. For a number of neurodegenerative diseases, things started to pop up in the literature here and there that these neurodegenerative diseases probably have a lot of lysosomal malfunction to them. And what really put it down was this article published in New England Journal of Medicine back in 2009 by Ellen Sidransky. She did what people had done before her, but she was rather clever. So this association had failed recognition in all of these genome-wide association studies that just had been run through Parkinson's, Alzheimer's, and were big at this time. But Ellen knew that Gaucher patients, and she saw a lot in her clinic, Gaucher patients were developing Parkinsonian symptoms. It was very, very clear. So she decided to look at an Israeli cohort and then further on a full cohort in Mass General to look at Parkinson's patients and see whether they in fact had different GBA mutations or glucocerebrosidase mutations. And they did. Glucocerebrosidase is now the most prevalent risk factor for developing Parkinson's disease, and that's widely accepted. In the meanwhile, the field had been flourishing because of this. Genzyme had discovered ceridase and developed that. Along had Shire had come, and another company called Protalix that had developed medicines, essentially the same, enzyme replacement therapies, recombinant proteins for this disease. But at the same time, there's also been an influx and about 20 years of understanding of the molecular mechanisms of glucocerebrosidase deficiency. Now this paper came out, and all of a sudden you can see, wow, there's something called Parkinson's disease, in which you have the exact same dysfunction. You have dysfunctional glucocerebrosidase. It's only the enzyme activity levels, roughly speaking, that actually matter. If you have below 20%, you start to develop Gaucher's disease in various cases, and the lower you get, the more aggravated the disease will become. If you lie something between 30 and 60% residual activity of glucocerebrosidase, you will most likely develop Parkinson's disease because there is a negative feedback loop between alpha-synuclein accumulation and glucocerebrosidase. Of course, the data had been confirmed in later large-scale meta-analysis, and recently, Michael J. Fox Foundation, which is a rather fantastic foundation, I haven't got any money from them, but they're doing a lot of good work. If you want to see how you can really move things forward as a patient organization, it's a good place to go and look. But they have basically defined and put GBA, or glucocerebrosidase, as it's called right here, as the lead hit for developing new therapies for Parkinson's disease. They have programs running now together with the industry that are moving things forward with that as their sole focus. That was basically taken understanding. So I hope you can follow it. You took something that came from cancer and understand lysosomal storage diseases. You can also go the other way around because you understand the molecular basis of disease. And of course, the fields evolve with you. And just be aware of that because then opportunities like this will arise. Unfortunately, in Orphazyme, we have drugs that can be used to treat glucocerebrosidase deficiency because we are focusing on Gaucher's disease. And now maybe we can also do something later on in life with Parkinson's. To come back to the theme of today, the future of life science, I think the only major topic I have is that common will become rare, or we will try to define it as rare. And the reason for that is that understanding of diseases at the molecular level basically provides the best foundation for rational drug development. That, combined with personalized genetic health and mass sequencing, will lead to our understanding of particularly neurodegenerative diseases and diseases that until now have been characterized by symptoms will to a much larger extent be characterized by the biochemical makeup and what molecular pathways are involved in diseases. The challenge to that is that people are looking to do this actively. Particularly, I mean, the spearhead of this has been cancer and oncology development, where a number of, if you go to patient conferences and speak with patients, they are extremely well informed, particularly in these rare diseases where doctors often are not that well informed. People, and particularly patients, their parents and the carers are reading up on this, and they're demanding knowledge and understanding on a molecular basis of what the disease means to them and to the children. Essentially, everybody's aligned towards this. Patient organizations want to see this happen, companies do it, regulators want to see it happen as well, because there's a number of good reasons for that, we can come into that later. Academia clinicians and payers want to see this happen as well. So everybody wants to understand this. The big challenge over the next 10 years as this happens will be pricing. Because as I told, the market is sort of in a schism right now, and if you follow the news about Valeant and Martin Shkreli, people are not too happy about this very, very free pricing, not even for rare disease drugs. That being said, if you look at the overall budget, and $400,000 per year for Soliris sounds like a lot of money, but overall, at least in the UK, in the overall health budget, treatments, even though they are exorbitantly priced for rare diseases, only cover about 2% of the total budget, or UK health budget. So that's also something you need to consider. And when you see prices like $400,000 per year, it does sound rather intimidating. But you also have to keep that in mind. This is for very rare, very life-threatening diseases where nothing else is being developed. The other side of that, of course, is that people will seek to exploit that, as everybody will always exploit a place where you can take economical advantage of a system. But it will happen, and we'll also see a pressure on pricing due to this. I think that's it. That's sort of where I can see future of life science going. And I hope you could see where we came from with Orphazyme. going from something common and rather boring, cancer, into lysosomal storage diseases. And basically, you can also use that to go the other way around. That's it. Thank you.